So in this lab, you're going to be taking a look at some slides that have regular cells or normal cells of that tissue, and on the other well, there will be cancer cells of the same tissue. And what you're looking for is, one, what happens when the cell cycle doesn't function as intended, and two, if the cells are cancerous, what are some features that people look at who do this for a living to determine whether or not a cell is indeed cancerous. So you're going to have a bunch of pipettes. You're going to have some chemicals. One of them is going to be a blue one, which is called a methyl blue solution. Another one is going to be called eosin, which is a reddish solution. And then we're also going to have something called a rehydration buffer. There is a second step if we want to do that, which requires a mounting medium, and that is where we would take cover slips and put those on top of the slides. I found that we don't need to do the cover slips, so it's an unnecessary step. So the directions that you are going to have in your lab are very similar, if not exactly the same, as the directions that are with the kit. It'll also have the pictures, which is very helpful in showing you images of what each step actually looks like. But I thought it would be helpful if I put just a very short video together to avoid any confusion. So you're going to take a slide, and for this video, I'm going to use slides that have already been stained, just so that we don't waste the materials. On the slide, it's hard to see here, but you're going to want to take a look at what is the bottom and what is the top of the slide. A good way to determine that is to just use your fingernail and just gently move your fingernail over the outside edge of the well, and you'll feel a little ridge, and that'll determine that that is actually the top side. The part that does not have a ridge would actually be the bottom part, which is what's going to sit on top of the microscope. So all the chemicals are going to be placed on the top side, and that ridge will be enough to keep the chemicals from sliding off. So in the first step, it says, using a transfer pipette, cover each of the wells with rehydration buffer. And so the purpose of the rehydration buffer is to do that, to rehydrate the cells that are on the slide. So we're going to use a rehydration buffer. That's our rehydration buffer. And there is no perfect amount here. You're just going to take and probably put enough on there just so that the whole thing is nice and wet, and that would be rehydrated. And you're going to let that incubate for about 1 minute, and room temperature is sufficient for that. Step number 3, you're going to take that rehydration buffer and you're going to want to get that off. So we're going to rinse it. So everybody should have a beaker of distilled water, and then you just simply maybe shake a little bit of it off very quickly. Don't take and continue to move the slide in the water, because what will happen is if you do that continuously, you will eventually take off the cells that are on the slide. If you use a paper towel, you can kind of just get on the edge, and the wicking motion of the water will travel up to it. You don't want to touch too much of the slide, because again, if you do that, you might be moving some of the cells around. Step number 4, using a fresh transfer pipette, cover each well with methylene blue. So we're going to take this one, we're going to put it aside, and we're going to open up the methylene blue. You want to make sure you get yourself a new transfer pipette. So we got a new transfer pipette. We have our methylene blue. We'll take a little bit of that. Grab our slide. Here's our slide, and we're going to just drop the methylene blue onto our slide. And you're going to want to make sure that you've got enough on there that covers the entire slide. Again, careful not to use the pipette to swirl around, because you'll disrupt the cells that are on the slide. Incubate for at least 2 minutes. So we would allow this methylene blue to be on there for about 2 minutes. And then again, same thing, I would recommend maybe taking the excess and putting it somewhere so that way you don't have just stuff dripping off. You can see I've covered most of it. It's not the best job. I would have liked to have gotten more methylene blue there. And then I'm going to take and I'm going to just rinse. You can see the water came out pretty blue there. And don't be too worried about being perfect with this, but do the best that you can. In fact, you can kind of see now that the cells are actually kind of bluish in color. They've been stained somewhat blue. The reason for the staining being done in a certain order is that certain stains penetrate certain organelles and certain parts of the cell. And once one of those have been, penetrated with that particular stain, it may inhibit the other stain. So like, for example, if the red stain is used to do the inside of the cell and the blue stain maybe stains the cell membrane, it might make it hard for the red stain to pass through the cell membrane because that stain is in there. So you would do the red before the blue. In this case, blue is first. Number 6, we've rinsed. Number 7, using a fresh pipette, you're going to go ahead and cover the slide with the eosin solution. Same thing. Immediately rinse in this one. So the eosin does not stay very long. Put that on, it's a quick stain, and make sure you get that off as soon as you can. Your cell is pretty much stained as it should be. The next step would be to just air dry. Allow that to dry for a little bit, and then you are all set to go to look at your cells under the microscope. I recommend starting off with the low power, the 40x, move up to 100, and then move up to 400. And then on the pages that follow, you'll be asked to draw some illustrations and look for some identifying characteristics to see what you see as the difference between a normal cell and a cancerous cell.